Hi, my name is Naoyuki Kamatani, the CEO of Taka Pharma Company. We are now developing a very promising new drug, which is in a clinical stage. So please listen to me. Today I'm going to talk about ATP or energy enhancement for the treatment of Parkinson's disease, dementia, and mitochondrial disease. We have problems to be solved. Just as energy is a critical issue for the planet, it is also a critical issue for the human body. Mounting evidence suggests that Parkinson's disease, Alzheimer's disease, and mitochondrial disease are caused by cellular energy shortage. No disease-modifying drug is available for Parkinson's disease, and no effective oral drug is available for Alzheimer's disease, vascular dementia, or mitochondrial disease. An AI study from 5 US universities to analyze a vast amount of data, including GWAS, omics, and others, identified fevizostatin as the most effective drug among thousands of drugs for Alzheimer's disease. Huge clinical data from 7 million patients confirmed the effect. In addition, several other papers reported the dementia-suppressing effects of XOR inhibitors, including fevizostatin and allopurinol. In the left side panel, US researchers reported that fevizostatin and allopurinol dose-dependently suppress dementia by about 40%. In the right side panel, Korean researchers reported that fevizostatin suppressed dementia, including Alzheimer's disease and vascular dementia, by about 80%. In addition, a meta-analysis confirmed the results. This is a list of papers from various countries that reported the dementia-suppressing effects of XOR inhibitors, including allopurinol and fevizostatin. Therefore, XOR inhibitors surely suppress dementia, including Alzheimer's disease and vascular dementia. However, before all of those reports, we had patented them in a very clever way. Using our unique technologies, we discovered this effect and patented combination drugs, each containing an XOR inhibitor, including fevizostatin and inosine. The fact is that we integrated all the data, including genome, mRNA, protein, and others, and clarified the precise mechanisms of Parkinson's disease. I don't have time to explain all the rationales, but from the genomics point of view, about 30% of the genes responsible for Parkinson's disease are mitochondria related. Alpha-synuclein gene explains only 5%. Intensive analysis strongly suggests that the most upstream abnormality is ATP shortage due to mitochondrial dysfunction. ATP shortage leads to dysfunctions of proteasome and autophagy lysosome systems, and finally causes the accumulation of aggregated protein. Then how can we enhance ATP? ATP molecule is composed of the AMP part and the high energy part. The AMP part is enhanced if hyperxanthine increases. Hyperxanthine can be increased by either XOR inhibition or inosine supplementation. If the AMP part is enhanced, the high energy part is also enhanced, since more glucose goes to the high energy part. So the combination of an XOR inhibitor and inosine has a very powerful ATP enhancing effect. Each of our drugs is a combination of an XOR inhibitor, gout drug, and inosine. Hyperxanthine or IMP can be substituted for inosine. Our patents cover more than 9 combinations, but this slide shows the 3 major combinations. Patents were approved in Japan, US, China, EU, etc. Then we tested our hypothesis. We administered fevizostatin and inosine twice a day for 2 weeks to healthy subjects. Blood ATP and hyperxanthine indeed increased, as shown in this slide. We also showed that the combination of fevizostatin and inosine has a synergistic effect as well as reduced side effects. In the left side panel, we administered fevizostat or inosine alone and measured hypxanthine. Administration of each of them caused only a little increase in hypxanthine, but a dramatic increase was observed when they were administered together. In the right side panel, we measured uric acid. Inosine caused marked hyperuricemia, while fevizostat caused marked hypouricemia. However, when both of the compounds were administered together, they cancelled adverse events each other, so the combination resulted in both a synergistic effect and reduced side effects. Then we administered fevizostat and inosine to 2 mitochondrial disease patients for 2 weeks. In both cases, biomarkers were dramatically improved. In the left side panel, BNP decreased by 31% in a patient with mitochondrial cardiomyopathy. In the right side panel, insulinogenic index increased by 3.1-fold in a mitochondrial diabetes patient. Then we administered fevizostat and inosine to 26 patients with Parkinson's disease for 8 weeks. As a result, the primary endpoint MDS-UPDRS Part 3 was significantly improved. Recently, we published a paper. Fevizostat protected mitochondria from age-related degradation and elongated the lifespan of C. elegans. Therefore, our drug may be effective for anti-aging. The market for our drug is huge. For example, the number of patients with Parkinson's disease is 1.5 million in US, Japan, and EU only, but it will increase to 30 million in 2030 worldwide. The numbers of Parkinson's disease and dementia patients will increase dramatically because of the aging of the population. First, we target Parkinson's disease because of the ease of designing clinical trials. This is the management team. I am the CEO, Naoyuki Kamatani. I authored over 600 papers, including 34 in Nature and Nature Genetics. Other team members are also reliable persons. Business plan, necessary expenses. We plan a phase 1 study this year and a phase 2 study next year. The cost required until 2025, when the phase 2 study finishes, will be $13 million. The phase 3 study will end in 2027, and the submission for approval will be in 2028. As soon as development funds are obtained, drugs for dementia and mitochondrial disease will be developed also. Business model, revenue model. The maximum sales will be about $800 million for Parkinson's disease only and Japan, US only. For exit strategy, IPO or M&A is contemplated when POC for Parkinson's disease is obtained in 2025 or upon approval in 2029. Enterprise value in 2029 is estimated at $720 million for Parkinson's disease only and Japan, US only. Some of you may be concerned about the pricing of a combination drug because this is a combination of 2 old compounds. But FDA approved Auvelity as a fast-acting antidepressant last August. Auvelity is also a combination of 2 very old compounds. However, the retail price of Auvelity is about $8,000 per year per person, which is pretty expensive. We received the FBRI Award at the 3rd Kyoto University Life Science Showcase in San Diego in 2022. Please let us know if you are interested in our ATP enhancers. Thank you very much for your attention.